spectrometry. Welcome.
Thank you. Hello everyone. Today I'm going to speak about the monitoring of 18 pollutants during treatment in a wastewater treatment plants by high-resolution mass spectrometry. Currently, a huge variety of organic compounds such as pesticides, drugs, or illicit drugs have been found in the aquatic environments, and one of the main sources of chemical contamination of aquatic environments is wastewater treatment plants because they receive and discharge a large number of compounds into surface waters. For instance, citalopram, an antidepressant, has been found in a wastewater treatment plant outlet with a concentration between 8 and 936 nanograms per liter. Some contaminants during wastewater treatment can be removed mainly by physical, chemical, or biological pathway. For example, they can be biodegraded or photolyzed. Other pollutants cannot be eliminated during this treatment, so they persist and get discharged into the surface waters, so they contaminated it. And for others, degradation leads to transformation products. So during a wastewater treatment, one compound may follow multiple reaction pathways and generate a large number of unknown transformation products. Nowadays, it is essential to study these transformation products in order to characterize the chemical contamination of our aquatic ecosystem. Indeed, a large number of transformation products is generated from one single compound, since one molecule can give n transformation products. Moreover, in some cases, they are more persistent and are more toxic than the parent compound from which they are derived. And finally, as I said before, a large number of those compounds remains unknown. So, the objectives of this work were first to determine tracer TP associated with 80 contaminants. Secondly, to characterize the fate of those 18 contaminants during wastewater treatment. And third, to qualify the impact of treatment processes of wastewater treatment plants using new tools such as non-target analysis or suspect screening. To characterize those transformation products and the contamination of aquatic environments, different analytical methods can be used. The first one is a very specific technique, and it is called target analysis, and its aim is to look for a short list of compounds in the samples. Then the identity of the compound is confirmed with the internal standards, and the quantity of the molecules can be obtained. The second one is a very specific method, but not only, because depending on its application, it can be quantitative, qualitative, or semi-quantitative, and its aim is to search for an extended list of compounds in the sample again, and then the identity of some candidates can be validated with the standards according to several parameters, such as the mass, the retention time, or the MS2 spectra. The last one is called non-target analysis, and it is a very qualitative technique. During this mode, all the masses are detected without a priori knowledge in the samples. Then, fragmentation pattern is used to obtain potential structures, and like suspect screening, the identity of some candidate can be validated with the standards. Moreover, for those two modes, the identity of the molecules can also be validated with the level of Szymanski shown here. For instance, level 1, which is higher, is obtained when the structure is confirmed by the analytical standard. In this work, we chose to work on 6 French wastewater treatment plants, so a total of 12 waters were sampled. The study sites are shown on this map. As you can see, several types of wastewater treatment plants with different treatment capacities and with various treatment processes were selected. So after that, 24-hour average composite samples were collected and then filtered. A solid phase extraction was performed on the sample using HLB Oasis cartridges, and then 3 microliters of the extract were injected and analyzed with LC cutoff. Before sample analysis, we must define a list of compounds to be studied, so we chose to work on 18 organic contaminants, including pesticides, surfactants, artificial sweeteners, phytoestrogens, and drugs. They were selected based on their environmental relevance, of course, and their physico-chemical properties. After that, A second list was created, and it contains, of course, 18 native compounds, but also transformation products from the literature and transformation products from an Excel spreadsheet developed using the EWAG in silico biodegradation model. The number of transformation products associated with each parent compound is shown here. So as you can see, more than 1,000 compounds were studied in this work and compiled in a specific database. After that, suspect screening was performed using TQ-TOF on our samples, and data dependence acquisition or auto-MSMS analysis was performed. So compounds were specifically chosen from the list elaborated earlier and were then fragmented. This mode allows us to obtain fragmentation spectra that are compared with those of spectral databases, so the identity of some compounds can be validated with a level of 2, which is one of the higher level of confidence. Now let's move, it's time for the results. Here are represented the number of detected entities, and as a reminder, more than 1,000 compounds were searched during the suspect screening. And as you can see, a high number of entities is detecting according to the site and the type of wastewater. Now, if we focus on the identification of the compounds, for the 18 native compounds, between 8 to 12 parent compounds were detected in the wastewater treatment plants. They were identified with a level of 2. And among them, 8 compounds were identified in both raw and treated waters of the 6 wastewater treatment plants, so they were persistent. And for example, we can cite venlafaxine, carbamazepine, and acebutolol. Concerning transformation products, 33 to 59 potential transformation products were detected, again depending on the wastewater treatment plant, and they were identified with a level between 2 and 3. Concerning persistent transformation products present both in raw and treated water, the number varied to 12 to 26. And for example, we can mention one metabolite of venlafaxine or desmethyl venlafaxine. Now, let's move on the number of potential CP detected only in treated water. This number range from 11 to 36. And for instance, we can mention one metabolite of carbamazepine, acridone. To compare the impact of treatment processes on the behavior of contaminant and transformation product, chemical fingerprints were realized. And here are represented those of the site 2 and the site 5. As a reminder, the site 2 applied one-stage biofiltration and the site 5 activated sludge plus biofiltration. In terms of TP removal efficiency, the two treatment processes seemed to be very similar because 26 transformation products were detected only in the raw water, so they seem to be eliminated by those two treatment processes. However, in terms of TP formations, they were different. 11 transformation products were detected only in the treated water of the site 2, which means that they were probably generated during the one-stage biofiltration. In the site 5, a higher number of transformation products were detected only in the treated water. And again, they seem to be generated by activated sludge plus biofiltration. In summary, the presence of organic compounds and transformation products was demonstrated in wastewater treatment plant during this work. Moreover, the formation of transformation products during wastewater treatment was also observed. Various treatment processes were investigated in order to evaluate their impact on the behavior of organic contaminants and transformation products. Finally, it must be stated that a large part of transformation products remain unknown, so for some of them, the standards are available. Buying them and injecting those analytical standards will allow us to better characterize, better identify, and possibly to quantify some transformation product, which is essential for the preservation of our aquatic ecosystem. Now, I would like to thank my PhD supervisor, Hélène Budzinski, my colleagues, and the partners associated with the project, and you for your attention.